Okay, let's talk about phototransduction. Phototransduction. Cool. So, in your eyeball, there are 3 layers to the retina. So your eyeball kind of looks like this. You got like the cornea, and then you got that part, optic nerve. Okay, that's your eyeball. This is the front. This is where the light hits. So the light comes in, and it hits the back of the eyeball first. And fun fact, when the light is being taken in, the lens, since it's a convex lens, it actually flips the image upside down. And then when your brain takes that information in through the optic nerve, because the optic nerve travels to the brain has to flip that information right side up again. And I think that's just fascinating. Anyhow, so right around the eyeball, all around, is your retina. And there's this part of the retina called the macula lutea. And the macula lutea is like your fine vision. Or your focused vision. And right in the middle of the macula lutea is the fovea centralis. And so, like, if that's your macula lutea, that was a terrible circle, then right in the middle of it, that's your fovea centralis. And the fovea centralis, that's like your super fine vision. So if you put your hand up to your face and focus on it, you'll notice like you're able to focus and then everything else is blurry. And then if you focus on something over there, like everything else around it besides that thing is blurry. So all the blurry parts you're seeing are all around here, all around the rest of the retina. That's like your sides and everything. And the macula lutea and the fovea centralis, it's like what you're focusing on in the moment. So, phototransduction is the process of taking this light and bringing it back to the brain. So there's 3 layers of the retina. So it kind of looks like this. So like I said before, this part right here, I'll put it in red. This part right around here, that's your retina. And it has 3 layers. So layer 1 looks like this. Okay, this is a ganglionic cell. Ganglionic cell. And this side of the ganglionic cell goes all the way back to the optic nerve. Optic nerve. Here's the optic nerve right here. Optic nerve. So the ganglionic cell, it's the first layer that the light will pass. The light does not interact with the ganglionic cell directly because the light goes all the way back to the layer that's furthest back. Don't ask me why that is. It's kind of strange. And then the second layer you have is this right here, which is called the bipolar cell. Bipolar cell. And that helps to communicate information between the ganglionic cell and our third layer, which is the rod. So this right here is your rod. Rods and cones they're next to each other, so cones are also part of this third area. And rods, since they detect light, and cones detect color. But mostly in this class we kind of focus more on rods. So light comes in, it goes like this. It goes past the ganglionic cell, past the bipolar cell, and hits the rod. And when it hits the rod, some changes occur inside of the rod that cause a signal to be sent to the optic nerve. So now let's talk about what changes those are that occur. So I'm going to go ahead and erase this beautiful diagram of the eyeball, all of these lovely terms. Pretty much on the surface of your rod, let's go ahead and blow this up. So this is on the surface of your rod right here. It looks like this. Now, this is during when it is dark. So on your rod, you have a few different things. First of all, you have something called opsin. This blue thing is opsin. And then there's this red thing in there. And the red thing is called retinol. This type of retinol is 11-cis retinol because it is bent. So that is 11-cis retinol. Now together, these two things, I'll do purple because red plus blue is purple. So together, they make something called rhodopsin. That's the opsin plus the retinol. It's called rhodopsin. Okay, next thing is right here. This is called PDE, or phosphodiesterase. You also have transducin, which is just an alpha, beta, and gamma on the bottom of the rhodopsin. So that's called transducin. That's supposed to be a D. Oh my goodness. Transducin. And then you have a ligand-gated sodium channel. So now sodium wants to come in here. And the ligand is going to be cyclic GMP. So I'm running out of colors real quick. Let's go with pink for cyclic GMP. Cyclic GMP. So kind of what it looks like is you got your receptor, and cyclic GMP is right in there. Okay, so that is what the surface of the rod looks like when things are dark. So when things are dark, positives are being let into the rod like that. And when the positives are getting let into the cell, then it causes an IPSP. to be released. And IPSP means it's inhibitory, so no signal gets sent to the optic nerve, nothing. And that's how you know it's dark. Now you may be wondering, well, Savannah, you just showed me all these fancy new names, and it looks like they're not even really doing anything. So what's going on with that? Well, I will tell you. Basically what happens is once it gets light again, okay, so this is in the light. Light comes, it comes over to the rhodopsin, and it strikes on to the retinol, and it turns the 11-cis retinol into, wait for it, straightens it out, it straightens out the bent, and now it's called all trans retinol. So if you remember when we learned about fats, the cis bond, the cis fat is bent and the trans fat is straight. So same thing, trans is straight and cis is bent. So all trans retinol is straight because the light came, it hit it, straightened it out. So this causes the alpha on transducin to actually move. Beta is still there. So alpha comes and moves over to phosphodiesterase, and it activates phosphodiesterase. Bing! And then phosphodiesterase is going to come over to the cyclic GMP and cleave the C off of GMP, which makes it so it can no longer bind onto the sodium gated channel. So now sodium cannot get in because this gate is now closed. So now sodium's like, what? Can't get in. So as a result, no IPSP will be sent. So there's no IPSP now. Oh, well, I've lost half of our bipolar cell. There we go. Okay, so now the bipolar cell by default releases an EPSP onto the ganglionic cell like that. And so that is going to make a signal, an action potential, be sent to the optic nerve, which then goes to the brain and tells you, hey, we're seeing over here, seeing something. So yeah, that's really it on phototransduction. So go ahead and rewatch this video as many times as you need, or kind of repeat it in your head as many times as you need, but it's not too much more complicated than that, thank goodness. And good luck.